In 1395, children in France began dying, not from the plague. The plague had already run its course by then, not from starvation. Children died from something that had no name. First, they stopped growing. Then, a strange lethargy set in. Not painful, but a deep-seated ache, as if a factory inside the child was running out. Then, skin problems began. It cracked, became inflamed around the mouth, and peeled on the hands. Their eyes lost their sharpness. In the dark, these children could barely see at all. Doctors of the time, if they could even be called doctors, attributed it to bad air, melancholy bile, and divine punishment. No one could explain why the disease specifically targeted children from poor families, specifically in certain areas, specifically where the staple diet was bread, beans, and porridge. Meat was a rarity. 500 years passed. 500 years passed before medicine finally figured out what was killing these children. And the answer turned out to be so simple that it's unsettling to think how many generations died waiting for it. They lacked a metal, or to be precise, 2 grams of metal. Not iron, that would be a familiar story about anemia. A metal that most people think of like this: batteries, pipes, something about galvanized roofs. Element number 30, symbol Zn. Grayish blue, brittle at room temperature. That's zinc. Welcome to Fool's Gold, a channel where we take elements from the periodic table and tell their real story, not the one in the textbook, but the one beyond it. And zinc is a perfect example. In the textbook, they write about it as, well, nothing special. But this is an illusion that crumbles as soon as you learn that without these 2 grams, your DNA stops copying properly. The immune system collapses, skin cracks, eyes go blind in the dark, children stop growing. That's where we'll start. Not with atomic weight or how zinc was born in the depths of dying stars. I know you're getting tired of this information, but we can't do without it. Let's start with a mystery that has plagued people for 500 years, and which no one has been able to solve. The Middle Ages, Persia, Egypt, India, China. Each of these civilizations had its own versions of the same picture. Children with stunted growth, adults with unhealed wounds, people who see normally during the day but walk into walls at dusk. And everywhere, a pattern that no one could decipher. This happened to those who ate little meat. The connection seemed obvious after the fact. At the time, it wasn't, because meat was scarce due to poverty, and poverty itself breeds disease. How can you isolate one cause from 100? Almost another 100 years pass. It's 1961. An Iranian doctor named Ananda Prasad is working in rural Iran. He sees young men, 20-25 years old, with the build of teenagers. They're 5'5" tall. Their livers and spleens are enlarged. Puberty has either not yet begun or is significantly delayed. Blood tests are more or less normal. Their diet consists of flatbread, beans, and a few vegetables. There's almost no meat. Prasad starts measuring everything and stumbles upon zinc. The zinc levels in these men's blood are meager, very low. He suggests a simple experiment: give them zinc supplements and see what happens. His colleagues initially refused to take what happened next seriously. The grown men began to grow, literally grow. They gained 10-15 centimeters in a few months. The body simply waited. He waited 5, 10, sometimes 20 years for a single substance, and once he obtained it, he triggered all the developmental processes that should have occurred in adolescence. It was so unexpected that the first publications of the study were met with hostility. But a single trace element can't have such a significant impact. That's too simple. Other factors must be involved. They searched for other factors for a long time, but found none. They found zinc. But why these men? Why rural Iran? Why the poor neighborhoods of medieval France? Why, as it would later become clear, does a significant portion of the planet's population still live with a chronic deficiency of this metal? The answer: food chemistry. More precisely, one specific substance found in grains and legumes, phytic acid. It's found in wheat, rye, corn, rice, lentils, and nutri. That is, the basis of the diet of most of humanity throughout history. Phytic acid is the storage form of phosphorus in plants. Plants need it, humans don't. And it has an unpleasant quirk. It binds zinc ions in the intestines, forming insoluble complexes. Zinc is present in food, but the body doesn't see it or absorb it. It literally passes it by. Meat doesn't do this. Zinc is readily available in meat. Oysters contain enormous amounts of zinc, about 75 milligrams per 100 grams, with a daily requirement of 10 to 11 milligrams. One oyster is a week's dose. This is why those who ate bread and porridge got sick. Not because their diet lacked zinc. Technically, it was there because phytic acid converted the zinc into an inaccessible form. This is where the story of how humanity accidentally discovered a solution to this problem begins. And this is where it began, and used it for thousands of years, without understanding why. Sourdough bread, a traditional bread with a long fermentation period, is used. During the fermentation process, bacteria produce the enzyme phytase, which breaks down phytic acid. The availability of zinc from sourdough bread is significantly higher than from quick, unleavened bread. People have been making sourdough bread for thousands of years, not because they knew about phytic acid and the bioavailability of zinc, but simply because that's what their ancestors did. When industrial baking switched to rapid-fire yeast in the 20th century and reduced fermentation time from 12 hours to 90 minutes, no one thought about zinc. They thought about efficiency, about standardization, about bread being uniform, soft, and staying fresh for a long time. Zinc got lost in this transition. Quietly, unnoticed. Today, zinc deficiency is one of the most common forms of nutritional deficiency in the world. The World Health Organization estimates that approximately 2 billion people suffer from it to some degree. 2 billion is more than the populations of China and the United States combined. A 500-year-old mystery. 2 grams of metal, 2 billion people. The phenomenon was not immediately recognized. The scientific community accepted its data with great reluctance. The claim that a single element influences growth, immunity, vision, wound healing, and a dozen other processes simultaneously seemed too radical. But biochemists weren't sitting idly by while the clinical debate raged. They dissected enzymes, molecule by molecule, and found zinc in every third one. Burt Valley of Harvard systematically studied metals in enzymes in the 1950s and 1960s. Carbonic anhydrase, the enzyme that transfers carbon dioxide from tissues to the blood, contains zinc. Without it, it doesn't work. Alcohol dehydrogenase, the enzyme that breaks down alcohol in your liver, contains zinc. DNA polymerase, the enzyme that copies your DNA with every cell division, contains zinc. Then the list grew faster than anyone could write it down. By the end of the 20th century, more than 300 enzymes were counted, in which zinc is not an impurity, but a working part of the molecule, a structural element. Remove zinc, and the enzyme falls apart. Millions of cell divisions are occurring in your body every second right now. Each time, DNA polymerase unwinds the DNA strand, reads it, and makes an exact copy. This mechanism works only because a zinc ion resides in the enzyme's active site. This zinc ion coordinates the nucleotides, ensuring accurate copying. Without this ion, the mechanism makes mistakes, or simply stalls millions of times per second. Zinc. The most elegant discovery in zinc biology isn't enzymes. It's what was discovered in the 1980s, when scientists learned to look at proteins in three-dimensional resolution. Proteins that control genes, so-called transcription factors, must physically grasp the DNA strand to turn the desired gene on or off. To do this, they require a special structural element, something like a clothespin. This structural element turned out to be loops of the amino acid chain, held together by a zinc ion. When the zinc ion coordinates 4 specific amino acids, cysteine and histone, in different combinations, the loop takes on a rigid, finger-like shape. They're literally called zinc fingers. Transcription factors use these fingers to wrap around the DNA double helix and read the genetic code. They decide whether to turn this gene on or that gene off. Depending on which fingers and where in the DNA they're attached, About 3,000 proteins in the human genome contain zinc fingers. 3,000. This is the largest family of regulatory proteins in our genome. They control embryonic development, the immune system, cellular responses to stress, and neuronal maturation. And they all rely on zinc ions. Remove the zinc, and the fingers will relax, and gene regulation will collapse. That's why zinc deficiency has so many manifestations. It's not just one function that's disrupted. Hundreds of regulatory points simultaneously begin to malfunction. Growth, immunity, vision, skin, wound healing, the nervous system, reproductive function, everything. Because zinc fingers are everywhere. And speaking of plants, zinc isn't just necessary for us. It's essential for normal photosynthesis. It's a component of enzymes that regulate chlorophyll production. Plants without zinc turn yellow, stunt growth, and stop absorbing carbon dioxide properly. Zinc deficiency in soil is one of the most common problems in agricultural lands worldwide. Children in medieval France starved not only because their diet lacked meat, but also because the grain they used to bake bread grew in zinc-deficient soils. It was a vicious cycle with no escape because no one knew what to look for. There's a paradox I want you to keep in mind. Brass, the yellow metal used to make doorknobs, bullet casings, and musical instruments, is a metal used to make brass. The trumpet in an orchestra is brass. The lock on your door is most likely brass. Coins are often brass. Brass is a medicine alloy, and it was made 2,000 years before anyone even realized zinc existed as a separate metal. This isn't a slip of the tongue. It's literally true. Roman coins from the 10th century BC, the sesterces and dupondii, were minted from orichalcum, as the Romans called it. It's a beautiful word. It sounds almost like gold. Orichalcum is brass in composition. Copper and zinc in a ratio of approximately 70-70. Roman coin masters knew how to produce it reliably, reproducibly, on an industrial scale. They had never even seen zinc in their lives. That's why zinc melts at 41 to 19 degrees Celsius. This is normal, a perfectly acceptable temperature for a metallurgical furnace, but it boils at 907 degrees Celsius, and the temperature in an ancient smelting furnace easily exceeded 1,000 degrees Celsius. This means that the zinc, barely having time to smelt from the ore, immediately evaporated, flew away, literally turning into steam and escaping through the chimney. It was physically impossible to detect it. By the time the furnace cooled, there was no zinc inside. There was a white powder on the walls, zinc oxide. The Greeks called it pampholics, or cadmium. Alchemists would later call it philosopher's wool. Pampholics, a beautiful name for something completely incomprehensible. But how then was brass made? After all, zinc is in it, so it must have gotten there somehow. It got there through the ore. Copper ore was heated together with zinc ore, calcite, which is zinc carbonate, and coal. In this process, the zinc was reduced from the ore, converted into steam, and this steam dissolved directly in the liquid copper. The zinc was converted into an alloy, bypassing the liquid metal stage. No one saw it. It simply ended up inside. For 2,000 years, humanity worked with zinc without knowing it existed. The first to figure out the trick weren't the Greeks, Romans, or Arabs, though Arab alchemists were quite accomplished. The first were metallurgists from Rajasthan, in the Zawar district of what is now Rajasthan, India. Sometime in the 1st century AD, perhaps a little earlier, local craftsmen figured out how to capture zinc before it escaped. The problem was simple. Zinc evaporates, so we need to capture it as vapor and cool it before it meets the air. Meeting the air is oxidation. It's a white powder again, not a metal. The solution they came up with is called downward distillation. It works like this. A retort containing ore and coal is installed, upside down. Beneath it is a cooled receiver, sealed to prevent air from entering. The zinc evaporated. The steam flowed downwards, not upwards, as usual, but downwards. According to the plant's design, it entered a cold, closed chamber, and there condensed into liquid metal, pure zinc, without oxygen, without oxidation. It sounds simple, but to come up with this, to reverse the entire conventional order, to build the plant upside down, this required unconventional thinking and a great deal of trial and error. Excavations in Zawar answered the question of scale. Archaeologists found about 9 million kilograms of spent ore there, along with thousands of used retorts. This wasn't a small workshop, it was an industrial operation. Zinc flows from the plants in quantities European chemists couldn't even dream of. The ore itself deserves special mention. The main mineral from which zinc is obtained is sphalerite, or zinc sulfite, ZNS. Black, heavy, with a characteristic luster. It was this mineral that was processed in Zawar. This is what is mined today at the world's largest deposits, in Australia's Mount Isa, in the Peruvian Andes, and in the Canadian mines of northern Ontario. Sphalerite is the entry point. Everything begins with it. At that time, Europe had no idea what was happening in Rajasthan. There was almost no contact between these worlds. Knowledge of zinc smelting moved slowly through Arab traders, through Persia, through long chains of transmission in which details were lost. In Europe, the first person to write about zinc as a separate metal was Paracelsus, around 1526. Paracelsus is a story in itself, deserving its own video. A scandalous, rude, and brilliant Swiss physician who publicly burned the books of Galen and Avicenna in protest against outdated medicine, who treated syphilis in miners in the Tyrol, and wrote the first ever treatise on occupational diseases of miners, who was hated by his colleagues and respected by his patients, who was the first to formulate a principle that is still relevant today. Everything is poison, and everything is medicine. The difference is in the dose. This man wrote something like this about zinc. There is a metal. It is similar to tin, but brittle, and it seems to be its own thing. Zinkum, that's what he called it. The origin of this word is still debated. One theory is that it comes from the German zinc, meaning sharp tooth, referring to the characteristic shape of zinc crystals when they harden. Another theory is that it is a distorted Persian or Arabic word that came via trade routes. But Paracelsus merely recorded a fact. There was no systematic understanding. What this metal was, where it came from, and how to obtain it remained unclear for another 200 years. Clarity came in 1746. German chemist Andreas Sigismund Margrave, the same one who would later discover sucrose in beets and prove its chemical identity with cane sugar, thus laying the foundations of the sugar industry, conducted a clean, reproducible experiment. He reduced zinc from zinc oxide under controlled conditions, measured its properties, compared it with other metals, and published a detailed description. This was the first time European science recorded zinc as a separate, independent element with specific, measurable characteristics. Margrave is considered the official discoverer of zinc in European science. However, it had already been produced in Rajasthan for 7-8 centuries. This is not a criticism of European science. It is simply indicative. A discovery is not always a matter of being the first. Sometimes a discovery is the first to explain and write in a language that later becomes universal. Margrave didn't discover zinc. He gave it a place in the system of knowledge. 54 years after Margrave, zinc changed the history of humankind, and in a completely unexpected way. 1800. Alessandro Volta, an Italian physicist from Como. He stacks alternating metal discs on a table, zinc, copper, and places rags soaked in a saline solution between them, connects the ends with a wire, and obtains an electric current. The first ever chemical source of direct current, the voltaic pile. The first battery. Why zinc and copper? It's no coincidence. The galvanic cell is based on the difference in the chemical activity of the two metals. Zinc readily gives up electrons. It is located significantly to the left of copper in the electrochemical activity series. This means that when paired with copper, zinc will always be an anode. That is, it will dissolve, giving up electrons. Copper, on the other hand, is a cathode, receiving these electrons. Electrons travel along an external circuit from zinc to copper. This is electric current. Volta demonstrated his invention to Napoleon Bonaparte in Paris in 1801. Napoleon personally witnessed the experiment. According to eyewitnesses, he was genuinely amazed. He immediately established a special commission of France's best scientists. It included Laplace, Monche, and Bertolli, and awarded Volta an annual pension and the Legion of Honor. A political gesture, of course, but also an understanding of its importance. Napoleon knew where history was turning, and the turn was significant. The voltaic pile gave scientists something they had never had before, a stable, controllable source of electric current. Before this, electricity was either static, lead jars, rubbed amber and glass, or atmospheric lightning. Neither could be used in the laboratory for long-term experiments. Now they could, and within just a few years, discoveries began pouring in. In 1800, William Nicholson and Anthony Carlisle separated water into hydrogen and oxygen using electrolysis, 6 weeks after discovering the voltaic pile. In 1807, Henry Davy isolated metallic potassium and sodium, elements no one had isolated before, using electrolysis. In 1808, barium, strontium, calcium, and magnesium followed. This entire series of discoveries was made possible by a stable current, and a stable current was possible because Volta used zinc and copper. Zinc is at the beginning of this chain. Quietly, unnoticed, as usual. But zinc's industrial history isn't limited to batteries. There are applications that affect literally every one of us. We just don't think about them. Look at any metal structure around you. A bridge, a fence, the roof of a neighboring house. If it's made of steel and not painted, it's most likely galvanized, coated with a thin layer of zinc. Why is this important? Because iron rusts. Everyone knows that. But not everyone knows what happens when a scratch appears on a galvanized surface. That's where things get interesting. A regular paint coating acts as a barrier. As long as it's intact, the metal is protected. A scratch appears, water reaches the metal, and rust begins. Barrier protection only works as long as the barrier is intact. Zinc works differently. It's more electrochemically active than iron, just as it's more active than copper when paired with it in a battery. This means that when water reaches a scratch where both iron and zinc are exposed, the zinc oxidizes, not the iron. Zinc sacrifices itself, taking on the corrosion. As long as zinc remains around the scratch, the iron underneath doesn't rust. It's as if the zinc shields the iron with its own body. This is called cathodic protection. And this principle was understood and applied long before anyone formally described it. The practical method of hot-dip galvanizing was patented in 1836 by the French engineer Stanislas Sarel. The steel part was immersed in a bath of molten zinc at approximately 450 degrees Celsius. A layer of several sublayers formed on the surface. First, an iron-zinc alloy, then pure zinc on the outside. The coating is durable, bonded to the substrate through a metallurgical bond, not simply glued on top. The service life of galvanized steel under normal conditions is 50 to 70 years. In aggressive environments, it's shorter, but still several times longer than non-metallic steel. Today, zinc-coated steel accounts for more than half of Meritzinc's total production, approximately 6 to 7 million tons per year. This includes bridges, rails, power line supports, car bodies, and building structures. All of this is based, in part, on the fact that zinc reacts more readily with oxygen and water than iron. The same property that made it the ideal anode in the first battery also makes it the ideal protector for steel. One metal, one fundamental property, two completely different applications. And here's another thing you probably didn't think about when you slathered on sunscreen before the beach in the summer. Oxyzinc, that same white powder from the walls of ancient smelting furnaces. The same Pomfilix that was given beautiful and incomprehensible names. Today, it's in every other sunscreen. It physically dissipates ultraviolet radiation without chemically reacting with the skin. That's why dermatologists especially recommend it for children and people with sensitive skin. For 2 years, this powder was a mystery. Now it's in your beach bag. There's one episode in this industrial history that deserves a separate mention. It's short but revealing. World War I, the Western Front. Trenches, barbed wire, artillery shelling. The barbed wire, by the way, was almost all galvanized. Otherwise, in the Belgian and French climates, it would have rotted away in a single season. Zinc was also there, albeit in a different context, but that's not the point. Military medics noticed a strange pattern. Wounded men with similar injuries healed at vastly different rates. The difference wasn't explained by age, the severity of the injury, or the quality of treatment. They then began looking at their diet before conscription, and it turned out that those who grew up in families where meat, fish, and eggs were regularly eaten healed faster. Those whose pre-war diet consisted primarily of bread and potatoes healed more slowly. This was then attributed to overall nutrition. Protein, calories, vitamins, it all made sense. Zinc was identified as a specific cause much later, after World War II, during systematic research in the 1950s and 1960s. It turned out that wound healing is essentially emergency construction. Fibroblasts produce collagen. Skin cells divide and close the wound. Immune cells attack bacteria. All these processes require active cell division, and cell division requires functioning DNA polymerase, and DNA polymerase requires zinc. Without zinc, construction slows down, wounds heal more slowly, and infections get a head start. Today, zinc oxide is an ingredient in countless medical ointments for burns, bed sores, and chronic cysts. This is the same white powder, the pomphylix of the ancient Greeks, the wool of philosophers and medieval alchemists, which settled on the walls of furnaces and was given beautiful, incomprehensible names. The same powder is now in a pharmacy tube. For 2,500 years, the same substance. It was used without knowing what it did, and when they learned, they began to use it even more. There are things that are destroyed quietly, not by explosion or catastrophe, but by slow accumulation, day after day, year after year, until it is too late to change anything. Zinc is one such case, only in reverse. Wait, I'll explain everything. In the 1970s, oysters in the North Sea began to die. Not all at once, gradually. At first, farmers noticed that the shells had become deformed, then they stopped reproducing properly, and then they simply started dying. Entire oyster farms along the coasts of France and Britain suffered losses. Fishermen complained. Scientists came to investigate. Water analysis revealed elevated zinc levels. The source was industrial waste, metallurgical plants, galvanic factories, and zinc ore mines. For years, all of these companies had been dumping zinc-containing waste into rivers, which carried it out to sea. And then they discovered something that few people think about when talking about zinc. Zinc is a biologically essential element for humans, fish, oysters, plants, and all living things, but it is necessary in certain concentrations. Any more than that, and it becomes toxic. And toxic precisely because it is biologically active. It competes with other metals for enzyme sites, displacing copper, manganese, and calcium from proteins where they belong. Mechanisms that should be functioning fail. Oysters are especially sensitive. They filter water and accumulate metals in concentrations that can be thousands of times higher than those found in the environment. This is called bioaccumulation. Oysters are like sponges for zinc. A single oyster can contain up to several hundred milligrams of zinc per 100 grams of body weight. For comparison, the food standard in most countries is around 50 milligrams per kilogram. In polluted areas, oysters accumulated zinc in quantities that made them unfit for consumption, and the oysters themselves simply couldn't function properly at such concentrations. The reproductive system was the first to fail. The story of the North Sea oysters is a localized episode, but it reflects a global problem that environmentalists began to discuss loudly around the same time. The Industrial Revolution of the 10th and 10th centuries saturated the biosphere with zinc. Not catastrophically, like mercury in a mortar, but systemically and ubiquitously. Main sources. The first source is the mining industry. Global zinc production currently amounts to approximately 13 million tons per year. The largest producers are China, Australia, Peru, and India. During the mining and processing of zinc ore, some of the metal inevitably ends up in waste dumps, wastewater, and dust. The second source is galvanized structures. Zinc is gradually washed off the surface by rain, especially in the first few years after coating. From roofs, fences, and bridges, it ends up in storm drains, and from there, in rivers. Third, rubber. Zinc oxide is a component of car tires as a vulcanizing agent. When tires wear down the road, rubber particles containing zinc settle on the asphalt and are washed down the drain. According to estimates by the European Chemicals Agency, ECHA, approximately 100,000 tons of zinc are released into the European environment annually. In Europe alone, in just 1 year. What happens to it next? Zinc accumulates in the soil. Plants are especially keen to absorb it. Some species, so-called hyperaccumulators, are capable of accumulating zinc in their leaves at concentrations that would be lethal to most other plants. The record holder is
Tlaspi carlescens, a humble herb from the cruciferous family. It can accumulate up to 30,000 milligrams of zinc per kilogram of dry weight in its leaves. This is approximately 100 times more than ordinary plants. Scientists initially dismissed this as a curiosity, but then realized it could be used. Phytoremediation. Cleaning contaminated soils with plants. You plant Tlaspi carlescens in a zinc-contaminated area. It grows, accumulating the metal in its leaves. Then you mow it down and dispose of the biomass. Repeat for several years, and the soil gradually becomes clean. It's not a quick process. It can take years, sometimes decades. But it works without chemicals, without heavy machinery, and without huge expenditures. It's simply a plant that evolution has taught to do something that took humans centuries to figure out how to use. Why can the gillworm accumulate zinc without dying? It turns out it's learned to sequester zinc in special cellular compartments, preventing it from reacting with enzymes, a sort of intracellular container for the toxicant. The mechanism is complex and not fully understood, but the fact remains: the plant has found a way to live with something that kills others. Now let's talk about the aquatic environment in more detail, because here the story becomes more complex and interesting. Zinc exists in water in different forms. The free Zn² plus ion is the most toxic. Zinc bound to organic matter is significantly less toxic. This means that the toxicity of zinc in water depends not only on its concentration, but also on the chemical composition of the water. In soft water with low organic content, the same level of zinc will kill fish. In hard water with a high organic content, the same concentration would be harmless. This poses a serious problem for regulators. You can't simply legislate for a maximum permissible zinc concentration of a certain number of milligrams per liter and call it a day. Water hardness, pH, and organic content must be taken into account. The European Water Framework Directive introduced the concept of the biotic legend model, BLM, which calculates the actual toxicity of zinc taking all these parameters into account. This is significantly more complex than simply measuring the concentration, but significantly more accurate. For fish, the most sensitive stages are eggs and fry. Adult fish can tolerate zinc concentrations that kill trout. Salmon, trout, and grayling, typical inhabitants of fast-flowing, clean, soft-water rivers, are particularly sensitive. It's no coincidence that in areas of active mining, rivers often empty of these species first. There are documented cases in Wales on the River Riddle near the old Metal Maze lead-zinc mines. Mining ceased there in the early 20th century, but acidic drainage water from the old mines continues to leak into the river, bringing zinc and lead with it for over 100 years. There are almost no fish in the river. Recovery is underway, but very slowly, because the source of the pollution has not gone away. The mines are not working, but they continue to leak. This is an important point. Zinc pollution doesn't end with production. Waste dumps, tailings ponds, old mines, they continue to leach metals into the environment for decades and centuries after closure. The legacy of mining is more than just a hole in the ground. They are slow-moving chemical reactors that operate non-stop. But here's what's important to remember. Zinc is not mercury. This is a fundamental distinction, and it's worth discussing separately. Mercury accumulates in food chains. Each subsequent level contains more mercury. Plankton, small fish, large predatory fish, the concentration increases with each step. This is biomagnification. Zinc doesn't behave this way. Living organisms are able to regulate the zinc content in their tissues. If there's too much zinc in the diet, the body reduces its absorption and increases its excretion. Accumulation up the food chain doesn't occur. A predator doesn't contain more zinc than its prey. This means zinc pollution is a serious local problem in industrial zones, but not a global threat like mercury in the world's oceans. The second difference is that zinc decomposes. More precisely, it doesn't decompose in the chemical sense. Zinc atoms are eternal, but zinc compounds in soil and water gradually transform into less accessible, less toxic forms. They bind with organic matter, clay, and carbonates, becoming geochemically inert. This process takes years and decades, but it does happen. Mercury doesn't. It circulates from the atmosphere to the ocean, from the ocean to plankton, from plankton to fish, from fish to you. The cycle is closed and practically endless. Zinc, in this sense, is a less hopeless story. Nevertheless, the problem is real, and it has specific geographical locations on the map that have become symbols of what happens when industry ignores the consequences for too long. Poland, Silesia, an industrial region that has mined zinc and lead for centuries. The town of Bukovina, near Olkusz, is home to one of Europe's largest zinc and lead smelters. It has been in operation since the 1960s. The soils around the smelter contain zinc in concentrations tens and hundreds of times higher than background levels. Vegetation in the surrounding areas is stunted. Some areas look almost like a lunar landscape: sparse grass, deformed shrubs, bare earth between them. But here's the interesting thing. It was in Silesia, in these contaminated areas, that Polish botanists discovered unique plant populations adapted to high zinc concentrations. Over the course of several hundred years, this is nothing by evolutionary standards. Some species have developed a resistance to the metal. Populations of bentgrass and Capillaris scalyx growing on zinc waste heaps are genetically distinct from populations of the same species growing several kilometers away on clean soil. They are less productive, but they survive where others perish. Evolution in real time. Humans contaminated the soil with metal. Nature responded. Slowly, reluctantly, with losses, but it responded. Now about another scale. Not local, but global. Zinc is involved in the global biogeochemical cycle. It sounds complicated, but the essence is simple. Zinc constantly moves from soil to plants, from plants to animals, from animals back to soil through decomposition, from land to rivers and seas, from marine sediments through geological processes, and back to land after millions of years. This cycle existed before humans and will continue after. We have merely accelerated some parts of it and slowed others. Industry has extracted zinc from deep geological horizons and distributed it across the planet's surface much faster than geology ever did. This has altered local concentrations in soils, rivers, and coastal zones. Globally, the cycle hasn't been broken, but regional ecosystems in industrial zones have felt the effects. Marine organisms are reacting in an interesting way. Phytoplankton, the foundation of marine food webs, uses zinc to fuel carbonic anhydride, the very same thing that transports CO2. In areas of the world's oceans that are linked by zinc, and such zones exist in the open ocean, far from continental sources, phytoplankton productivity is lower. There's less phytoplankton, less fish, less of everything else. Experiments enriching seawater with zinc in laboratory conditions have shown increased phytoplankton productivity. This theoretically opens the question of how to manage marine ecosystems. But this is an area where science is currently observing rather than intervening. The system is too complex, and the consequences of any intervention are too unpredictable. I'll conclude this section with something that I think will surprise you. Zinc is one of the few metals where environmental concerns and biological necessity are literally on the same scale. Too little is a disease, and too much is a disease. Moreover, the boundary between too little and too much depends on the form in which the metal is present, the chemistry of the environment, and the species. This makes it unlike mercury or lead, where the logic is unambiguous: the less, the better. That won't work with zinc. You can't simply say, let's get rid of zinc in the environment. Because without zinc, there won't be normal phytoplankton, there won't be normally growing plants, there won't be normally functioning animals and people. A balance is needed, a precise, delicate balance that depends on the context. Nature has maintained this balance for billions of years without our help. Over 200 years of industry, we've seriously undermined it. Now we're figuring out how to fix it, preferably without breaking anything else. This, frankly, is a pretty good description of what ecology as a science does. Let's get back to the frog. Not the one in the biology textbook, but a specific one. A dead frog lay on a metal table in Bologna in the autumn of 1780. Luigi Galvani, an Italian anatomist and professor at the University of Bologna, was dissecting a frog. It was a typical workday. Metal instruments lay nearby on the table. At one point, the scalpel touched the sciatic nerve of a dissected frog's leg. At the same time, the leg came into contact with a zinc instrument on the table. The leg twitched. A dead leg, severed, without a brain, without a heart, without any source of life. It twitched. Galvani was shocked. He repeated the experiment. It twitched again. He began experimenting systematically, hanging frog legs on copper hooks attached to the iron railing of a balcony. The legs twitched in the wind, touching the railing. Galvani decided he had discovered animal electricity, a special life force stored in the nerves and muscles of animals. He was wrong in his explanation, but the observation was correct. Alexander Volta, having learned of Galvani's experiments, objected. There is no special animal electricity. The frog is simply a conductor. Electricity arises from the contact of two different metals, copper and the iron of the railing. The frog here is only a salt bridge between them, like a wet rag in his future battery. The dispute between Galvani and Volta lasted several years. Both were right in some ways. Galvani correctly saw that muscles respond to an electrical impulse. This is the basis of all neurophysiology. Volta correctly understood that the source of current is the contact of metals and built a battery. Zinc is at the center of both discoveries. In the experiment, Galvani is one of the metals creating a potential difference. In the battery, volts are like an anoth, giving up electrons. But here's what is rarely mentioned. The dispute between Galvani and Volta sowed a seed that sprouted 20 years later in a completely different place. 1818. London. A young woman, 20 years, publishes a novel. A novel about a scientist who creates a living creature from dead matter using electricity. Her name was Mary Shelley. The novel was called Frankenstein, or The Modern Prometheus. Mary Shelley grew up in a circle of people who animatedly discussed Galvani's experiments and the controversy surrounding animal electricity. The idea that an electrical impulse could make dead flesh move, which is exactly what Galvani did with frogs. And this is exactly what Galvani's nephew, Giovanni Aldini, repeated in 1803 on an executed criminal in London, passing an electric current from a voltaic pile through the corpse. This idea was in the air and formed the basis of the main science fiction novel of the 19th century. The sun in Volta's battery, Galvani's dead frogs, Aldini's public experiments on corpses in London, Mary Shelley, Frankenstein. Such is the chain, from metal on a lab bench to a monster assembled from dead bodies. After 20 years and a few casual conversations. But let's return from literature to science, because in the 20th century, zinc did something that is rarely mentioned in popular texts, and which is directly related to how modern medicine works. 1980s. Molecular biology was undergoing a revolution. Scientists had learned not only to observe genes, but also to read them, copy them, and transfer them from one organism to another. Genetic engineering was taking its first steps, and here a problem arose. To change the function of a gene, to turn it on, turn it off, strengthen it, weaken it, it was necessary to find a way to precisely and predictably affect a specific section of DNA. Not the entire molecule at once, but precisely the desired fragment. This was a problem that biologists did not know how to solve. In 1985, British biochemist Aaron Cluck, together with colleagues Jonathan Miller and Andrew McLaughlin, described the structure of proteins that do exactly this in nature. They sit precisely on a specific section of DNA and controls the gene. These were the very same zinc fingers we talked about earlier. Loops of the protein chain held together by a zinc ion. Cluck had already won the Nobel Prize in Chemistry by that time. He received it in 1982 for completely different work, for the development of crystallographic electron microscopy. Zinc fingers are a separate chapter of his biography, in no way connected with that prize. He was simply one of those people who made important discoveries regularly. Kluck realized that if nature uses these structures for precise DNA recognition, then it was possible to create artificial zinc fingers tuned to any desired sequence. Assemble them into a chain and get a molecular tool that will find exactly the right section in the genome and do something specific there. This is how zinc finger nucleases, ZFN, were born. Artificial proteins that find a specific section of DNA and cut it. After the cut, the cell triggers a repair mechanism, repairing the DNA. And so at this point you can insert a new fragment of genetic code into the cell, which will integrate itself in place of the cut. This was the first truly working genome editing tool. CRISPR-9, which is on everyone's lips now, was still almost 30 years away. ZFNs appeared earlier and paved the way. Clinical trials of ZFNs began in the 2000s. One of the first objects of application for HIV. The logic is as follows. HIV enters the cells of the immune system through the CCR5 receptor. If this receptor is damaged or disabled, the virus cannot enter the cell. A small number of people have a natural mutation in the CCR5 gene. They are called delta 32, and these people are effectively immune to most strains of HIV. Using ZFNs, scientists began to do the same thing artificially. They take the patient's immune cells, turn off the CCR5 gene in them using zinc fingers, and return the cells back. Clinical trials were conducted at the University of Pennsylvania under the direction of Carl June. The results are mixed, but in principle, the method worked. In a number of patients, the modified cells survived and multiplied. It's not a cure for HIV, but it was the first real step toward editing the genome of living people, using a zinc ion as a tool, holding together a protein loop. Today, the FN has been largely superseded by CRISPR, which is simpler, cheaper, and more flexible. But without zinc fingers and the work of Klug and his colleagues, this whole history of genome editing would have begun much later, or would have taken a completely different path. There's another story I want to tell. It's short, but it's about how science sometimes stumbles upon an answer from a completely unexpected direction. Alzheimer's disease is one of the most studied and yet least understood diseases of our time. A characteristic symptom is the accumulation of amyloid plaques in the brain, a cluster of beta-amyloid protein which should normally be eliminated from the brain, but in Alzheimer's disease clumps together into insoluble aggregates. In 1994, Australian neurobiologist Ashley Bush and his colleagues discovered something strange. Beta-amyloid. It aggregates significantly faster in the presence of zinc ions. Zinc literally provokes blindness. Add zinc to a test tube of beta-amyloid solution, and it begins to flake before your eyes. This immediately gave rise to a hypothesis. Perhaps Alzheimer's disease is partly a disease of impaired zinc metabolism in the brain? Perhaps zinc dysregulation is part of the mechanism. Research is still ongoing. The picture has turned out to be much more complex than it seemed in 1994. Zinc performs vital functions in the brain. It accumulates in synaptic vesicles and is released during nerve impulse transmission, regulating neuronal activity. This is a normal, necessary process. The problem, apparently, is not zinc itself, but rather the disruption of its transport and distribution mechanisms in the aging brain. But the very fact that zinc has become the focus of research into Alzheimer's disease, a disease that currently affects approximately 55 million people worldwide, a number that is growing as the planet's population ages, says something important. Zinc isn't a peripheral element of biology. It's at the very center. In cell division, in gene regulation, in synapse function, in the immune response, in wound healing. Take it away, and everything falls apart. Let's put it all together now, but not the usual way, not with a list of facts or a solemn final paragraph. That's what personally captivates me most about this story. Zinc has been with humans from the very beginning. In coins, in jewelry, in brass tools, in sourdough bread that people baked intuitively. in oysters that they ate and didn't know why they felt better. In the ointments they used to treat wounds, there was zinc oxide. They just called it by other names. In the sunscreen powder that the Egyptians rubbed on their skin, long before anyone thought of writing SPF on a tube. Humanity has used zinc for thousands of years, not knowing it existed. Then it was recognized, given a name, placed in the periodic table as number 30, and began to be mined industrially. It was used in batteries, in galvanized structures, in zinc die casting. It is used to make carburetor bodies, door fittings, hundreds of small parts that you hold in your hands every day without thinking about the composition. It figured out enzymes, zinc fingers, cell division, and in the process polluted the soil and rivers around industrial zones, destroyed oyster farms in the North Sea, and left behind slow-flowing chemical waste that will leach metal into the river for centuries to come. Knowledge hasn't made us more careful, it has made us more efficient, including in causing damage. This is not pessimism, it's just an observation. There is one question that I deliberately put off, the simplest and most inconvenient. Where does zinc come from on Earth? I'll tell you briefly, as I promised at the beginning. Zinc is a product of nuclear reactions during explosions of massive stars. When a star several times heavier than our Sun reaches its end and explodes as a supernova, elements up to about number 40 or 50 in the table are synthesized in the shockwave. Zinc is number 30. It falls right in this range. Some zinc isotopes are formed during the process of silicon combustion in the depths of the dying star. Some at the moment of the explosion itself through the capture of neutrons in the shockwave. Gas field clouds enriched with these elements, billions of years later, became part of the protoplanetary disk of our Sun, from which the Earth formed with zinc inside. That's it, we'll say no more about stars. This is not the main thing in the history of zinc, something else is important. Here's a final thought experiment for you. Not scary, just accurate. Take an ordinary day, your day. In the morning you wake up, your brain has restored synaptic connections overnight, partially with the participation of zinc in the synapses. Have breakfast. If you eat meat or eggs, replenish your zinc reserves. If you eat only oatmeal, not really. You walk outside, cross a bridge, or drive on a road with guardrails covered in galvanized steel. You pick up a doorknob. It's probably brass, meaning copper and zinc. You open a lock, almost certainly the same. You use your phone. It has a battery that conceptually goes back to the voltaic pile, although now it's lithium. You put on sunscreen before a walk, zinc oxide on your skin, the same white powder from the walls of ancient ovens. You get sick, you take a cold pill with zinc, and maybe it shortens the illness a little, or maybe not. Science is still debating here. Inside you right now, 3,000 proteins with zinc fingers are reading your genome. 300 enzymes are waiting for zinc to do their work. DNA polymerase copies DNA millions of times per second in every dividing cell of your body. 2 grams of metal, grayish-bluish, brittle at room temperature, discovered by European science in 1746 and produced in India for centuries before that. A metal that was anonymous for 2,000 years, that was killing people with its absence slowly, imperceptibly, until a SAG measured the blood of Iranian peasants in 1961, that made a dead frog's leg twitch and thus started a chain that led to the battery, to electrochemistry, and to the novel about Frankenstein, which today stands at the center of Alzheimer's research and genome editing. Not flashy, not toxic in itself, not liquid, not radioactive, not precious, simply necessary everywhere and always. Quiet? The most important things rarely seem important. They seem ordinary. A galvanized fence, a battery and a remote control, 2 GMA in a body, and 500 years of children who did not grow up until someone finally thought to measure what they were missing. That's the whole story of zinc. Element number 30, unnoticed, irreplaceable, and in some ways the most human of all the elements, because its history is a story of how long we can ignore what we can't live without. But you know what still haunts me after all this? The periodic table has 118 elements, and each of them has the same story. Not a boring list of properties from a textbook, but detective stories, catastrophes, accidental discoveries, wars, mad scientists, and dying children. Each element has its own people, its own mistakes, its own moments, when everything could have gone completely differently. The next element we'll talk about is the lightest metal on the planet. So light that it floats on water. So reactive that it can only be stored in oil, otherwise it will ignite right in the air. But that's not the most interesting thing about its history. The most interesting thing is that 90 years ago, it was added to lemonade, sold in stores, drank by families, and advertised as a cure for hangovers and bad moods. And then it turned out that this light, a fizzy, pleasant drink, does something to the human psyche that no one expected. And this is just one of its stories. Have you guessed what element we're talking about yet? See you very soon on the Fool's Gold channel. Until then.